Good evening once again, and this is Emily Live Lessons. When we're going through Biology Paper 1 tonight, you could either be live here right now, or in the future, watching this recording. So as a whole, there are 4 units to this. So we have 1, 2, 3, and 4. So cell biology, organization, infection response, and bioenergetics. So these are the times I plan to spend maximum on each of these. And first we'll start off with cell biology. Now I've taken into account that each tier and group, as in combined science or triple science, will have different advanced information, and you'll see it on the bottom right on each slide. So overview for unit 1 will be cell structure, cell division, transport, like diffusion, and the required practical for some of you, microscopes. So first with the cell structure, we have 2 types of cells. First one is a prokaryotic cells. We have lots of simple cells, and these store the genetic information in strands or loops. So if we draw a little cell, at a minimum, it will have these are its strands, and at least a cell membrane, and the empty space, the cytoplasm. Whereas some more complex cells, these are called eukaryotic cells. And these instead store genetic information in a nucleus. So if we draw another cell, it will still have a cytoplasm and cell membrane, but rather you'll see this nucleus. And the function of DNA in both cells is just the same, just for instructions to make a new cell when it divides by mitosis, which we'll come to in a bit. Now we'll need to know some parts of a cell, and here's a big table of them. I suggest you should take a big screenshot of this and put this into flashcards. I'll add a few notes to this. For example, mitochondria, for example, are too big to fit inside the bacteria cell, so this is actually the cell wall, perhaps in the plants, are made from something called cellulose. The permanent vacuole also then keeps some dissolved salts right here, which links to osmosis, which we come again later to. Moving on, we've also got the cell structure of a bacterial cell, where you should know the purposes of each part. And however, I'll highlight a few bits in red. Like this. For example, the flagellum is to move easily. And the purpose of these bacterial cells is simply to divide, like especially in our body. So if you take another screenshot of this and take down a few notes, this would be good. Now, the first major thing we need to look at is the cell cycle. So cells need to replicate themselves because cells really just die of various things, whether that's infection, um, gamma rays, like those kind of things. They need to be replenished. Otherwise, we would simply die, the structures would fall apart, and so on. So the cell cycle includes mitosis, which is also called copying division. So let's say we have a cell, and in it we have our nucleus, and to simplify things, we've got two pairs of chromosomes, a red one and a green one. And let's say as well, it will have other things inside it, such as its mitochondria, ribosomes. So the main part of the cell cycle is that the cell replicates. So replicates two things. So the DNA, the chromosomes, which are actually right here, and all the organelles, or the subcellular structures. So these are like your mitochondria, the ribosomes, and so on. So what we eventually get is the same cell, still as one, we'll still have a nucleus, but eventually we'll get loads of chromosomes, 2 times as many as we need. And still we have 2 times as many of the organelles. So that's like, again, the mitochondria, ribosomes, and so on. Now this is where the actual mitosis starts. If you looked at a cell undergoing mitosis under a very fine microscope, first what you'd see is, in the cell, all the pairs of chromosomes line up in the middle. A bit like this. And you'd often see this little dashed line where they're all tied together, and then they're just kind of pulled apart. Now that we've got the correct number of chromosomes for each cell, like just a pair in each cell, the cell membrane divides, as well as the cytoplasm, to form two daughter cells. And what you can tell about these daughter cells are, they are almost identical to the parent and each other. This here, as you can see in the bottom right, is one of the major focuses of combined science, both foundation and higher tier. So to recap, the cell cycle, most of it is replicating the DNA and everything inside it as well, such as your mitochondria, ribosomes, and so on. And then the second part, which takes much less time, is the actual division. So that's your DNA lining up in the center, pulling apart, or your chromosomes rather, and the cell membrane. Next is for triple foundation and high students. This is about transport or diffusion, so the how things move. So by definition, which you'll need to know, it's the movement of molecules from an area of high concentration to an area of low concentration. So that could be, for example, a perfume sprayed in one corner of the room and it diffuses because it moves randomly and eventually spreads to an area of low concentration. So that's like everywhere else. Now, the thing is, there are things diffuse at different rates, and this could be because of, let's say, temperature. This is because higher temperature, higher kinetic energy. This is also a link to physics. So this is in the molecules. And therefore, it's simply put, they move faster. So this kind of speeds the process up. Another factor could be the surface area between the two areas. For example, if they have to pass through a membrane, it's better if it has a larger surface area. For example, these two membranes, so this will have a high surface area, whereas a more flat one like this will have a low surface area. And if I draw arrows where things can diffuse with little blue dots, you can see that this wavy form increases the area where things can diffuse rather than this on the flat surface. The last factor you need to think about as well is the concentration gradient. So that's a difference in concentration. So for example, if you've got an area of high concentration to an area of where there's practically none of the molecule, that's going to have a higher concentration gradient and therefore a higher rate of diffusion. So think about it like a slope. So you've got high to low. And this right here, the slope, is like how fast it diffuses or how fast it will even out. An example can be oxygen diffusing into cells while carbon dioxide diffuse out of the cell. Now, diffusion was important for the early organisms because the thing is, this happens randomly. This means it doesn't require energy. The problem with getting energy is you have to do stuff, such as killing other organisms and then digesting them. So that means they could obtain loads of resources they needed. So this could be anything like oxygen, even glucose, perhaps, and also get rid of anything they didn't want, such as CO2 from respiration. Now, here's a common exam question that can appear, perhaps. It could ask, perhaps, why small unicellular organisms, like that just live as individual cells, even bacteria, perhaps, whereas us, more complex organisms need structures like lungs. So what if we model cells into cubes, perhaps? So we've got a cube, and its length is basically the same around it. So for example, its volume is n cubed, with its surface area. So you have 6 faces, each with an area of n squared or l squared. 6l squared. Now, small organisms will have this very small side length. For example, let's say 5 micrometers. So that would equate to like 0.0000005 meters. And if you do the maths, its surface area is this, and the volume even smaller, because if you cube something small, it becomes very small. This means its surface area to volume ratio is 120, or rather 1,200,000 cubic centimeters, like square centimeters per cubic meter volume. So basically that means there's lots of area on the outside for stuff to diffuse in and not much inside. In fact, you could rather write this as a ratio. This could be like 1,200,000, much, much larger than bacteria. So let's say they have a side length of 5 centimeters. Now, if we do the maths, the volume is 5 cubed, which is 125 cubic centimeters. Surface area is going to be 6 times 5 squared, which is 150. So if we do the surface area to the volume ratio, which is 150 to 125, if you divide both sides by 25, you get the ratio 6 to 5, which eventually gets you down to 1.2 to 1. So this means per unit of volume, there's not much surface area around the mouse. So therefore, we can't just simply let it diffuse, let oxygen diffuse. This is what we explain here. So we've got the different surface area to volume ratio. So we can see the larger organisms simply don't have enough surface area for the large volumes, because the large volume will have several more cells, and these cells will need more oxygen, more glucose, will need to get rid of more carbon dioxide, more waste, and so on. And simply using diffusion isn't going to work, because it's going to travel far to get inside the cell, or there won't be enough simply to diffuse. So that's why these large organisms will have complex structures like lungs, so this to compensate for this low surface area. The other type of transport we need to look at is active transport. So diffusion only works to an extent until the two sides, or the sides of substances, have the same or similar concentration. So for example, at the start, in the small intestine, we could have a high concentration of food. So this, we're talking about, let's say, glucose, whereas a low concentration in the blood. So therefore, we got quite a high rate of diffusion. But however, later, Since substances are moving from this inside the intestine into the blood, if nothing happens, that concentration will decrease and become more like medium. Whereas inside the blood, since it's getting more of that food inside, that will also go to medium. So it rises from low to medium. So now we get no diffusion at all. And therefore, well, nothing else can get in. Now, active transport uses energy, as the name suggests, to get more of a substance into a cell, even though diffusion can't happen or it goes against the concentration gradient. So it could even work, for example, if we use the same diagram, if it's like in the intestine, like a low concentration of glucose to like a higher one inside the blood. So active transport will just use energy to still like kind of force it away. And a good example of this could be root hair cells using this to keep the concentration of mineral ions, such as magnesium, higher in the cells than in the soil, because they need to absorb as much of these mineral ions into the cell and use them elsewhere in the plant. Now, many of these cells that use active transport, let's say in the small intestine, will have more mitochondria. If you remember, mitochondria carry out aerobic respiration, which releases energy. And this energy is what can be used then for this active transport. So therefore, we've got more mitochondria, so you can have more mitochondria doing this aerobic respiration, so more energy can be released for a higher rate of active transport. Now, we're still on triple foundation and higher content, where they have to do a required practical and microscopes. So there's two parts to it. The first one is to prepare a slide. So for example, in this case, we're going to use onions. So if you remember, you had to get a very thin layer of the onion skin, or just like peeling it off. Trying to get it one cell thick or transparent. And you'd try and do this either with your fingernails if you've got them long enough, tweezers perhaps, maybe a scalpel. It's sometimes easier the first time perhaps. The next step in preparing the slide, before we look at it, is to lay the cells in the middle of the slide. So if I draw out the slide like this, so this is like a plastic um, rectangle that's transparent, and then I'll draw the onion just in the center, and you've got to lay it down thick so we don't trap as much air bubbles, especially on the bottom. So this is our little onion slice right here. Next, you have to lay the cover slide, so that's a thin piece of plastic, so that's like this. And carefully try not to get this on the table directly, because they're like as invisible as contact lenses. And what we do is, let's say we have the slide. What we start doing is laying that cover slide 45 degrees, and then we just kind of place it down carefully. So almost in like a pressing motion. We're just letting it topple down. And this is to make sure we don't get as many air bubbles trapped inside, which will make our image worse. And last, that's it. What we do then is we have the stage in the microscope, and we fix these little arms on. Then that's ready. Now when we use the microscope itself, we first start with the lowest magnification on the objective lens. So we have two lenses here. So one at the eyepiece, and this is usually times 10. And then what we'll actually have, sorry for the terrible drawing, is another lens. So this is the objective lens. And these, for example, can come in a few different magnifications, such as times 4, 10, even 40. And what happens is these two magnifications combine together. So if something is enlarged by 10 and then 4 again, in total, for example, that'll become times 40. And what we do is to bring the stage up to the lens, and with the smaller lenses, it won't touch, the slide won't touch the lens. So we bring the stage up to the lens. And then what we do is we then lower the stage again, slowly, until we get a nice clear image, for example, like this. So we'll get nice, almost rect strips of the cells, and you'll see, manage to see tiny dots, and you should probably sketch it out if you need to in the exam. And it'll be stained with iodine, which is a nice orange. Now if you take a screenshot of this, and we'll move on. Next is osmosis, and its definition is the movement of water from an area of high concentration of water to an area of low concentration of water across a partially permeable membrane. So basically this is like diffusion, but for water molecules. And like diffusion, it will be affected by the same factors, for example, temperature, surface area, and the concentration gradient. Next is the required practical with osmosis, and that's for also triple foundation and higher. And this is where we use the potato cylinders, if you remember. So first we need to multiply those, so prepare those potatoes, like, and you need to make them all identical, all the same length, mass if you can, and all the same cut. So like we want cylinders, not maybe bits stuck in. So this is to ensure that they have the same surface area. Next, you need to weigh the mass of each potato on a top pan balance, and then place each one into a single boiling tube. And remember, you need to record the mass of each, and perhaps label it on each boiling tube so you remember which one is which. And then you'll add 25 cubic centimeters of a sugar solution into each boiling tube, and in each boiling tube you'll have to use a different concentration of sugar. For example, in one you'll just have the concentration of 0, which is basically just distilled water or water, 0.1 moles per cubic decimeter, 0.2, and some others you could possibly use are 0.4, 0.8, 1, and so on. Next, you'll need to cover the top of the tubes with plastic, so this is to make sure no water evaporates. And finally, after you've left the potatoes for around 48 hours, you could leave them perhaps for another 24 hours. You get the potato tubes out and then dry them with paper towels to make sure you get rid of the excess water that's stuck on there. And so you can get the true mass of the potato now that its water has either moved in or moved out. And you'll calculate the percentage change of the mass, and then you'll draw a graph, which should look like this. So we use the change in mass on the y-axis and the solution concentration on the x-axis. On this diagram, you see where the curve of best fit or the line of best fit, whenever it reaches the or crosses the x-axis, the horizontal one, this tells you there was no change in mass, and therefore the same water concentration, because no movement of water means like the same water concentration. So randomly, the water molecules would move in and out. So if I model this, let's say with water, these could be the concentrations of sugar. And then since there's no difference, in concentration, there's no movement going about, therefore no osmosis happening. So that's when the concentration inside the potato is equal to the concentration of the water. And really you'd have to only estimate that from the graph. And don't forget your units. These concentrations can either be in moles per cubic decimeter, which is in chemistry, or grams per cubic decimeter. And then we're on to the next unit. So that is for animal tissues, organs, organ systems. This is an overview. And plant tissues as well. This is really for triples. First, for all groups and tiers, so that's both combined and triple science, we have to look at the digestive system. This is an example of an animal tissue, organ, and organ system. So you should probably know the organs involved, such as the mouth, stomach, liver, gallbladder, small intestine, and large intestine. You should be familiar with each of their purposes. And overall, as a system, they need to digest food. Digestion is where we break down insoluble large molecules into smaller molecules to absorb. So these can diffuse into blood. And we have two types of digestion, mechanical and chemical. So mechanical is often within the mouth. So if we label this with an M, so this is within the mouth and the stomach as well. Whereas chemical, this is with enzymes. This can happen in the stomach, perhaps in the small intestine, though really enzymes are produced here in the liver. Now, these enzymes themselves, which are a major focus within this kind of topic or unit, we have three of them. So first of them is carbohydrates. An example of one is amylase, and these digest carbohydrates into sugars, or rather glucose. So what you see is you'll have big chains of glucose all bonded together like this. And then you'll need to break each of the one down, and that's what this enzyme will do. Protease will digest proteins into amino acids, and again, proteins are just chains of amino acids joined together, and the enzymes will simply break these down. Lipase, these digest fats and oils into fatty acids and glycerol, and it's a good thing to get these down to flashcards as well. because you simply need to know this. Now, each of these enzymes are produced in a few places. Amylase is produced in the mouth, pancreas, and small intestine. Protease is produced in the stomach, pancreas, and small intestine. And lipase is produced in the pancreas and the small intestine. So really all three are produced in the pancreas and small intestine. Now we get to look at how enzymes actually work. So enzymes are biological catalysts. So these naturally speed up the rate of reaction or how fast something happens. And remember that, like other catalysts, these can only speed up one type of reaction, and we can explain this with a lock and key model. So suppose we can model some starch, like little squares. It's like a very long chain of glucose molecules. Now, what an enzyme will do, it will almost, perhaps, mark out the shape of these, then kind of punch a hole. So we have two bits of an enzyme here. So this is the enzyme here. And I'll highlight two parts. So this bit where this little starch molecules or glucose molecules, the thing that fits into the enzyme like a key into a lock, is called the substrate. Whereas the lock itself, where the substrate fits into, is called the active site. So basically, to summarize, only one substrate or key fit into one active site, lock. And we need to remember this. Now, enzymes work better in some conditions than others. So they work best depending on the temperature and pH around it. So first with temperature, this is also a link to chemistry. Generally, a higher temperature will lead to particles moving faster. This is also like the kinetic energy of the particles, so they'll move around faster and collide much more frequently. And that means, usually, you'd have a faster rate of reactions. However, these higher temperatures can cause the enzyme to denature, and to denature means to simply change shape permanently. So normally, on the previous slide, our little enzyme that digested starch or the carbohydrates looks like this. Let's say we'll call this normal, a normal enzyme. And the denatured one will look something more like, so it's kind of messed up, if you like. And the active site has changed, which I'll highlight in green. And when the active site changes, it can no longer fit the substrate it was designed for it to accept, if you like. So that means it can no longer split up those molecules into smaller ones, and that means this enzyme can't catalyze that reaction anymore. So it can't break down those starches, for example, or proteins, or lipids. Another factor that may affect the enzymes can be the pH. So if the pH is wrong or far from the optimum, again, the enzyme will denature. But this is because the enzyme, if we look at it carefully, there's actually a chain of amino acids, for example. And if there's the wrong pH, these amino acids will start to break their bonds and then change. And if it changes again, the enzyme becomes denatured. If the enzyme becomes denatured, the active site changes. It can no longer fit with the substrate and catalyze a reaction. One organ we need to look at in the digestive system is the stomach. Now, what does the stomach do? So it contains first the hydrochloric acid to kill the bacteria in the food or any other pathogen. This hydrochloric acid also provides the correct pH for pepsin. So this is one enzyme or one type of protease. As well as that, it does some other mechanical digestion. So this is where we physically move the food around. This is like churning. So where we kind of circulate around all the contents in the stomach. Next, we need to look at bile. And this can often be a 4-mark question. Now, bile is a special chemical and it's alkaline. Now, this mixes in with the stuff or partially digested products that leave the stomach. And if you remember, the stomach has hydrochloric acid, which is acidic. Now, the enzymes that work in the small intestine, they work at a higher pH or more alkaline or neutral pH. So if we put acid straight into the small intestine, those enzymes won't work and they won't digest the contents. So first, the alkaline property of this bile will neutralize the acid, so raise the pH, which is more optimum for enzymes. This is because especially we'll look at one type of enzyme, which is lipase. So these will digest the fats into fatty acids and glycerol, and these will work best in the small intestine in the neutral or alkaline conditions. The next thing is that bile emulsifies fats. If you study food technology or like cooking, you'll learn that to emulsify is to mix liquids that don't tend to mix together. An example of this could be water and oil. And what you get is that the fats will then break up into smaller droplets, just like water and oil, so you can see the oil hovering up in small droplets. As we saw with smaller things, they will have a larger surface area to volume ratio. So for example, have a large droplet of fat and then break it down, let's say, into 4. Then we use blue dashes again to see where enzymes could act on. You can see that breaking it down will have a higher surface area for enzymes to work on. If you have a higher surface area, you have a more higher chance or frequent chance of each molecule of fat colliding with the enzyme. And if it collides more, there's a higher rate of reaction and that gets digested more. That's again a link to chemistry. So overall, we've learned so far that the stomach and bile helps so with digestion. Another thing that this helps with digestion is the small intestine. And now there are a few features, perhaps loads, to talk about, and we're going to compress this into answering a 6-mark question. So we've got an image right here, and we need to explain how the small intestine is adapted to efficiently absorb digested products. Pause the video if you like. Now that you've tried this question, one of the first points you can make is that villi increase the surface area, like this little pattern here. So it kind of folds back on itself, and even then you've got microvilli. So each cell is kind of has its own villi like this, so it further increases the surface area again. The next thing you could look at is the rich blood supply. So this basically takes digested products immediately away. And what this does is that we maintain a high concentration gradient. Next thing you could look at is the villi are 1 cell thick. And if you got a short distance, so cells are like very small, it doesn't have much distance to travel or diffuse simply. Whereas if it was, let's say, more than 1 cell thick, it would have to travel through those cells, which is much slower and decreases the rate of diffusion. So the thinner the walls are, or the thinner that the villi are, the better. So it's a shorter distance to the blood supply. And lastly, you could say that there are many or more mitochondria in the microvilli. And if you remember, the mitochondria carry out aerobic respiration, which release more energy, which is then used for active transport. And finally, we look at the large intestine where its purpose is just simple, absorbing water. And after that, what's left or what isn't digested forms a stool and exits via the anus. That's when you poo. Now finally, we have a required practical for all tiers groups. So that's combined and triple science, foundation and higher tier. So these food tests are where we use something called a reagent. So this is a chemical added to the food samples often in the test tube. So you add a few drops of these special chemicals, and then you should often shake it and then see what happens to it. And usually it's something to do with color. And before all of these food tests, you need to prepare a food sample. So to prepare a food sample, you would often grind it with a mortar and pestle, and then dissolve it in distilled water. You could possibly as well filter out any insoluble solids, such as anything that's left, like skin. Now, for the food tests themselves, you'll need to learn four of them. The first one is glucose. The reagent used for this is Benedict's solution, and this is a nice light blue color. As said on the previous slide, you would usually add it to a test tube and shake it, but this has a special condition for it to be used. So you have to place it in a water bath or a beaker of hot water, which is about 75 degrees, for a few minutes. Now, positive result means that we find glucose in the sample, so the solution turns from blue into something like red, orange, yellow, or green. So this is in decreasing glucose concentration. So for example, brick red would be very high. Just a normal red would be more high. You could say orange is medium, yellow is a bit just below medium, then green is just low. If glucose isn't found, the solution remains blue as it is. Nothing will change after those 5 minutes. You will also need to learn the test for starch. In this case, we will use iodine. After you add a few drops to it and then perhaps agitate it or shake it in the test tube to your food sample, if starch is found, the solution turns a blue-black, like almost like this color. Whereas if starch isn't found, the solution remains orange, which is the color of this iodine. You may want to take a screenshot right now. Next, the third test you need to remember is for proteins. And for this, you'll have to use the reagent or chemical Bioenergetics solution. So this is a more pink, like or like a colorless one. Right, so positive result is where the solution will turn purple or pink. That's when protein is found. Otherwise, it just remains colorless. For fats and lipids, you test with a Sudan 3 solution. So this is more of an orange color. This is like similar, really, to like fats. More of like a dark orange, perhaps. If there's a positive result, you'll have this layer on top. So if I draw a test tube, a positive result would be the rest of your sample, and then a more nice orange layer on top. So this is where the fat all gathers. It's a bit like the oil floating on water. So this is where whereas the negative result, no layer forms, and it just remains it is. It'll just remain in tiny droplets. Now, for triple science higher, they'll need to know about plant structures. One of them could be in the leaf. From top to bottom, you have your waxy cuticle, upper epidermis, the palisade mesophyll, the spongy mesophyll, the lower epidermis, as well as your guard cells in the same bottom layer. You'll need to be familiar about their adaptations and how well they'll carry out what they're meant to do. So, for example, the waxy cuticle, this protects the leaf from predators. So it will often deter many animals from eating the leaf. The epidermis, doesn't have to have much, but it's also like another structural purpose as well. The palisade mesophyll, these contain the chloroplasts, and these chloroplasts will carry out photosynthesis. And usually these chloroplasts are concentrated near the top. So this is because they want to absorb more light, and light is needed for photosynthesis. This also means that these two layers must be transparent. The spongy mesophyll is where the gas exchange happens. So this is where it'll absorb carbon dioxide and then release oxygen. That's the products from photosynthesis. It might even release a bit of carbon dioxide because remember, all cells respire um, and will produce carbon dioxide as a result because the energy is needed for growth. And just like lungs or the villi in small intestines, this also spongy texture makes it have a large surface area. The last thing you need to know about is the guard cells. So these will open and close depending on the conditions outside. So these, for example, will limit water loss because of the transpiration stream, as well as gas exchange. Next, you need to know about some other plant structures, which are xylem and phloem. So xylem carry water, and these cells here are often dead cells, and these won't have any cytoplasm, for example. This is because what you don't want to happen, if you add a cytoplasm, it would have some solute dissolved inside, and then water would move into the cell by osmosis, and that would make the cell expand, become turgid, and just block the water coming up through the xylem. The phloem will carry sugar and other things. That'll go up and down. and water is just carried up because it's carried from the roots to the leaf, whereas sugar is carried to every rest of the plant. This is because glucose is needed everywhere else in the plant because every cell needs to respire. Now, the combined science higher tier, they'll need to know about the food tests and apply these. So they'll apply these to see the effect of pH on enzyme activity or the rate of reaction. So first, they'd measure out a set volume, like 10 cubic centimeters of the enzyme they want, and put it into a boiling tube into a water bath set to, let's say, 30 degrees. And then they'll add a buffer solution which changes the pH down to 3, for example. Remember, we're changing the pH, so this is the independent variable. Next, they'll also measure out the sample of food, let's say 15 cubic centimeters. They'll put it into a separate boiling tube because we don't want the food to be digested immediately. And they'll put it in the same water bath so it reaches the same temperature. So when they reach the same temperature, let's say after a few minutes, you'll then mix them together, and that's when the enzyme could perhaps start working in digesting the food. Then at regular intervals, you'll then perform the food test on a small sample on a dimple tray. So that's where we have small wells where we can put each of these food samples and do a mini food test on them to see if one product has been made or one product is slowly disappearing. And you stop testing at the first sample that shows the reaction is finished. So that's kind of like the time taken for the enzyme to digest all of, let's say, the carbohydrates, fats, or proteins. And you repeat this again, and we want to change the pH every time. So we'll use a different buffer solution that will change the pH instead to 5 or to 7 or to 9. And to visualize this, we'll use the enzyme amylase at different pH levels where this enzyme will digest starch into glucose, and each of those will have different food tests. So the first two steps was to put the contents of each boiling tube, so the enzyme in one test tube, and then the food sample in another test tube or boiling tube, and then into a water bath. And remember to wait 5 minutes so they reach the same temperature as the water bath, which is 30 degrees Celsius. Then you will combine the two contents together and immediately start a stopwatch. And then every, I'd say 30 seconds, you'd then take a small sample of the mixture using a pipette and you'll add it to one well. And for example, we're testing if starch is still present. If the enzyme digests the food sample and eventually digests all of it, no more starch will be left, and then we won't find starch. So the iodine that we add will stay orange. So we can see on the right, so if the iodine still turns blue or black, there's still starch, and therefore the food sample is still not digested, and the reaction isn't finished. Whereas if iodine stays orange, there is no starch anymore because it's been digested and the reaction is finished. So that's when we stop the timer. The rate of reaction is hard to calculate because we can't exactly measure how much starch there is, because these tests are what we call qualitative. So these tell us if something is in there or not. It's as simple as that. So rather, to calculate the rate, we have to do some arbitrary number such as 1000 divided by the time taken. You should probably take a note of that down as well. On to the next unit, we're going to look at infection response. So we start with diseases. So this is for combined science foundation and both triple science tiers, so foundation and higher for triple science. Now, communicable diseases are caused by pathogens and they can spread from one organism to another. So that's one term you need to know. Whereas non-communicable diseases aren't caused by pathogens and don't spread from one organism to another. Health is a term you'll hear often, and it's a state of both mental and physical well-being, not really just an absence of disease, because you could still not have disease and still have other conditions. And then the pathogens themselves are harmful microorganisms to us, and these want to use our body and help them to multiply, which is what any organism needs to do. So we've got a few disease examples. So first we have the pathogens and we have bacteria, and these are small living cells, and these are 100 times smaller than animal cells. If you remember, bacteria cells can't have mitochondria as well because they're smaller than mitochondria. It won't fit into them. And these small things will cause diseases like food poisoning, salmonella, gonorrhea, and so on. Viruses are even smaller than bacteria. And these will cause diseases like TMV, HIV, measles, and so on. And a good thing to note is that bacteria cells will divide outside cells, whereas viruses divide or replicate inside cells. These are how they cause cell damage and make you feel ill. So these bacteria, once they've reached a larger number, they'll also release toxins. Whereas the viruses, they'll keep replicating inside the cell because they use the cell itself to replicate, like the ribosomes. These will each cell will keep filling up with viruses until there's too many and it bursts. The last two microorganisms we need to look at are fungi, which are single-celled organisms, and they vary a lot. These cause diseases like rose black spot in plants, athlete's foot, and so on. And lastly, protists. These don't directly pass from one person to another, but these require a vector. So a vector is like something in between. For example, with malaria, it can spread from human to human, but via the mosquito. So from human to mosquito to another human. As I've said on the previous slide, bacteria cause illnesses because they release the toxins that harm cells. And remember, they can replicate quickly depending on the conditions. They can go replicate or double every 20 minutes. This is because of the warm temperature inside the body. Whereas viruses will multiply inside the cells because they'll use the ribosomes and make more copies of themselves because they inject the DNA or RNA and they'll burst to form just replicate inside the cells, they'll fill up the cell, and when there's too many, it will burst. And those high number of viruses, so there'll be like millions, those will then infect more cells and so on. Now, diseases can spread in different ways. One of the most obvious is direct contact. For example, athlete's foot, you wouldn't want to touch someone with athlete's foot because you would get the fungi that causes it, for example, from skin to skin contact. The next could be indirect contact. So let's say someone that is infected with any disease, like caused by pathogens, they could contaminate the surface by, for example, coughing, simply touching it, and spilling fluids and so on. And then if another person touches the same surface that isn't clean, they will also get the pathogen. Diseases can also spread by sharing bodily fluids. They can also spread by air droplets. And this is how many viruses and respiratory illnesses is like COVID would spread. And lastly, we've got the vectors, which is how the protists will spread. They'll carry the pathogen and usually aren't affected by you. Since we know how pathogens spread, we know ways to cut down this transmission. For example, being hygienic is a good option. For example, we avoid the skin to skin contact, that's the direct contact, or we clean surfaces that could be contaminated. That could be using disinfectant, soap, sanitizer, and so on. This could include your own hands as well. We could also isolate infected individuals to make sure they do not spread the pathogen from themselves to another. Lastly, you could also destroy the vectors. For example, with mosquitoes, you should really get rid of still water or stagnant water because that's where the mosquitoes multiply and breed. And if you take away that stagnant water, they can no longer breed, their population may eventually fall, and eventually they can't spread any more malaria. Now, how does the body fight against all these diseases? Firstly, we're going to look at the human immune system, and it defends us in two ways. Non-specific defense systems and specific ones. We'll start off with the non-specific ones. So the skin, for example, acts as a physical barrier, so it physically stops any pathogens coming into our body, because this is just a layer of dead cells. What you also have are the hairs and mucus in the trachea and bronchi, because when we breathe in air, that's air from the surroundings. That air can contain pathogens of all sorts, whether that's viruses and even bacteria. And this mucus, for example, that lines the trachea and bronchi, these will trap the dust that may contain pathogens, so it stops the pathogens from reaching other cells. Then the mucus is wafted up to the throat by ciliated cells. Like this. That's a little bits of mucus moving this way. It'll go up to the throat and then swallowed and then sent to the stomach, where we remember contains hydrochloric acid, which kills the pathogens. And also, in our bloods, we have some white blood cells, such as phagocytes, and these engulf pathogens. So if I draw one right now, so we've got one like this. This could engulf, like, example, a tiny bacteria. So it engulfs it, this will close, and then it will kill it with enzymes. And it will render bacteria useless, and eventually if we keep eating these bacteria, then we could be okay. However, this isn't very effective on its own. This is because the bacteria, if you look closely, will have these little hairs on. These make it very hard to grab. This is where some specific defense systems come in. Now, the first of them is antibodies. So these are also produced by other white blood cells. These could be called um, B cells or plasma cells. So these produce antibodies which attach to a pathogen's antigen. So an antigen is like a little marker, as you can see in the diagram, that will latch on to the antigen. So each antibody will specifically hook on or attach securely to one type of antigen. So that's depending on the bacteria or virus or disease. And then it makes it easier for other white blood cells to engulf, so it can grip harder perhaps. And lastly, we have antitoxins, which are produced by other white blood cells. These attach to bacteria toxins and neutralize them, so they stop them from harming the cells. Next, we come on to immunity. Now, after we fought off an infection, some of these white blood cells that produce antibodies or antitoxins, these will divide slowly by mitosis because mitosis is controlled. You don't want cells to rapidly divide very quickly because that would become cancerous cells. Now, some of these cells will become memory cells, and these memory cells will produce antibodies against the same pathogen if it infects again. And the antibodies will be produced at a faster rate, along with more of them. Perhaps immunity is quite necessary for it, so this is like an evolutionary advantage. Because what our bodies don't want to do is, well, fighting disease requires lots of energy. And what you don't want the body to do is to keep fighting the same war, perhaps. Otherwise, we'd run out of energy, we'd run out of resources that produce these antibodies, and it'd simply waste time again, and it could kill us. Now, immunity usually happens like this, natural immunity, when we're infected with the pathogen itself. But however, some pathogens are quite dangerous to cause serious illness with long-term effects or even death. So that's why we can use vaccinations. And this can be a high mark question to explain how vaccination makes us immune. This basically makes us immune, but without exposing you to the pathogen. This is because dead or attenuated forms of the pathogen are injected in the vaccine. So these dead or attenuated, so these weak pathogens won't do anything. They won't cause a disease. And a good thing to remember is that they'll have the same antigens as the real more deadly pathogen. So if there's enough of these pathogens, they'll stimulate our immune system. So they'll stimulate these white blood cells to produce antibodies against a pathogen. Remember, it'll produce the same antibodies against the real deadly pathogen. And then immunity is reached again. So once we've killed all these dead pathogens, so these white blood cells will then divide by mitosis and produce memory cells again. And then at reinfection, the memory cells activate antibodies produced at a faster rate. And usually we'd vaccinate a large number of the population if there are fewer side effects, as it reduces its chance of death, transmission, and even infection from disease. And if a large proportion of the population is vaccinated, others who aren't vaccinated can be protected themselves due to herd immunity. So suppose if a disease can even infect a vaccinated person, they're less likely to pass it on. And even then, if they do pass it on, they are more likely to pass it on to those who are vaccinated against the disease. So it's very hard for the disease to spread among the unvaccinated, perhaps. Now we move on to the types of drugs that try and fight against disease, a bit like vaccines. However, some drugs just counteract the disease someone has, such as by relieving the symptoms themselves. These are painkillers, and these don't act against the pathogen itself. Whereas others, like antibiotics, work against the pathogen itself. These will contain chemicals that work against the pathogen and kill it. Antibiotics will contain chemicals that will kill the bacteria. However, this is a common exam question. Antibiotics will not treat viral infections because remember, viruses infect the cells by going inside them, and it's hard to get a chemical or drug into the cell where viruses multiply without damaging the cell itself. Now, to test the drugs, the vaccines, any of those, even painkillers, they'll need to go under some testing, and this can be a high mark question. A 6 marker one, for example. So describe how a treatment for disease, such as painkillers or vaccines, are tested before being given to the public. So overall, we have two parts. We have the preclinical trials and then the clinical trials. So the preclinical trials start with single cells and tissues in a lab, such as from an animal or a human within a lab. Next, the drug is then tested on live whole animals in labs. So this finds the toxicity of the drug, so how harmful it is. If it's already harmful to single cells, how harmful is it going to be on whole organisms, like even humans, functioning humans? It's going to be more toxic. Same goes for step 2. If it's already toxic to animals, it will probably be toxic to humans as well. Usually, this is much better. For example, if the drug affects organ systems, this gives us better information, so more. And then we move on to the clinical trials themselves, so that when it's approved or okay to test in humans. So we first test them on healthy volunteers. So we test the toxicity of the drug again to make sure that if it's not harmful on healthy volunteers who don't have perhaps any disease. So a range of doses are tried. So they'll start very low and increase it, and at some point it will get harmful. Because what you don't want to do is have a low dose and have it being very toxic, because it's likely going to be more toxic on sick patients. And this brings on to the second phase of the clinical trials. These are on patients. So now we test if the drug actually works, or the efficacy, or how effective it is, to see if it works against a disease against pain, perhaps if it's a pain relief drug. And then, if possible, you could even test it on more volunteers, so a larger sample, on a wider range of population. So that's where we do a double-blind trial, so on 1,000 people. So they'll be divided into two groups. So we have a placebo group and a real group. So placebo, the ones in the placebo group will be given a fake version of the treatment. It looks like it probably tastes like it, but it just doesn't work. So this is almost controlled. So this controls the behavior. So for example, if there is a drug that perhaps somehow lowers weight, the placebo will make sure that um, behavior is accounted for. Because if you take a drug that lowers your weight magically, you'll probably exercise less, eat more unhealthy, and so on. And double blind means that the people administering the drugs as well don't know if they're giving the placebo or the real drug as well. This is important so they don't tell the patient either to change their behavior or influence their behavior. Now, for triple science in the higher tier, they'll need to know about monoclonal antibodies. This is often a high mark question where we describe how they are produced. So a definition is several copies of one type of antibody. Now, the problem is antibodies are produced from B cells, or the B lymphocyte cells. The problem is we know that they divide slowly, just like other cells, but even slower. And the process is we have to first use a mouse or rat. So we infect it with the pathogen we want, and they will produce antibodies or the B lymphocyte cells that produce the antibody we want. And these B lymphocyte cells would be found in the spleen, so we remove it. And then these B cells will be combined with the cancerous cells. So cancerous cells divide rapidly or multiply very quickly. That means we get these hybridoma cells. So they'll have the genes of the B lymphocyte cells, but will divide quickly. So what we've got now are several cells, all with the genetic material from the B cells, and they will all produce several antibodies. Now, a use of these monoclonal antibodies could be for pathogen testing. Those of you who were in the COVID pandemic knew that you had to test often, for example, using lateral flow tests. So these are stuck onto a sheet, and then we have a sample of, for example, something stuck up your nose from the swab, and then with a buffer solution. And then these antibodies will have a little dye stuck to them. In this case, it would be the line. So a positive test for, let's say, COVID would be like this. So if you remember, you had the sample well, and then a line next to a T, and then a line next to a C if it's positive. This is because if the sample, let's say, from your body contained the virus, it would attach to the antibodies at the T here, and then when they attach to the T there, and the fluorescent dye, such as the red dye, will stick to it and form a line. And lastly, we need just this C here, so that will just come up, should come up, regardless of a positive or negative test to show that the sample has travelled over. The last unit for tonight is photosynthesis, which is for combined foundation and higher, as well as triple science foundation tier. As a basic, you'll need to know that photosynthesis is used by plants to produce glucose, because glucose is needed in aerobic respiration to produce energy. Now, Photosynthesis uses light energy because it's endothermic, so it needs energy from somewhere for it to happen. And the word equation you need to know is this. Carbon dioxide plus water react together to form glucose and oxygen, just with the help of light energy. And you'll need to be familiar with the formulas of each. And I suggest you learn this formula for glucose and put this on a flashcard. Now, glucose could be used in a few ways. Firstly, they could be used for respiration. Just like other organisms, plant cells need to respire and use this energy for other things, such as to produce cellulose, which form the plant cell walls, to produce amino acids, because glucose can combine with nitrate ions, which are from the soil, to form amino acids. And if you remember, chains of amino acids produce proteins, and these proteins will have structures, such as a leaf or an organ. And this basically makes the plants grow. Lastly, they could be used for storage. So when they come to a time where they can't use photosynthesis as much, they can use the reserved glucose. However, this glucose needs to be stored as starch or as lipids or fats and oils. And this is a common exam question. Explain why it's an advantage for plants to store food as starch rather than glucose. You need 3 points for this. One of them first is that the fact that starch is insoluble while glucose is soluble. So starch will not dissolve in water. This is firstly a good thing because starch won't affect the concentration of water near cells, whereas glucose will. So if you got more glucose, you got a lower water concentration. And if this happens, the concentration of water is affected, osmosis can happen. For example, if the concentration of water is lower outside the cells because glucose is just hanging around there, water can be drawn out due to osmosis, causing the cell to become weak. That means the plant as a whole can become weak. Now, combined tier, foundation, and higher tier of combined science, they'll need to know about factors affecting photosynthesis. So this is basically almost anything it needs, really, such as water and carbon dioxide, because they are reactants. The amount of sunlight or light intensity will also affect the rate of photosynthesis, because light is needed. The temperature will also affect it, because photosynthesis involves enzymes. Because these enzymes will need to move a lot quicker, and these can collide together at a higher temperature more frequently. You also need to think about the amount of chlorophyll in a plant. So this is the pigment that's green, and this will absorb the light, and this light will be used to kick off photosynthesis. And deficiencies, so that's where the plants do not get enough, let's say, mineral ions such as magnesium. They won't produce as much chlorophyll, they won't absorb much light, so less photosynthesis. The last bit is the surface area of leaves exposed to sunlight. Now, a required practical for combined science foundation and higher tier would be light affecting photosynthesis. So we have our variables. First, our independent variable. So this is what we change. This is going to be the light intensity. Our dependent variable is what we measure, and that's the rate of photosynthesis. If we measure the rate of reaction, we can measure either the amount of something produced over a certain amount of time or the amount of something used up. It's probably better to measure the amount of oxygen produced because these are bubbles in water. And control variables will be kept the same, so make sure the test is valid, so the results are valid. So we know that one factor, the light intensity, will change the other factor, the rate of photosynthesis. This could be temperature, the carbon dioxide level, water level, same plant species, and so on. Now, we've got some equipment here. And these will have some measures in, for example, the water with the sodium bicarbonate to control some of the variables, such as this sodium bicarbonate here will control the amount of CO2 dissolved in the water, so it will remain the same. The temperature can be controlled by using an LED bulb or with a beaker with water. So what this will do, instead, the water absorbs any heat, whereas using an LED bulb is more efficient and is like less heat, so it gets less hot. And lastly, the light intensity can be changed by either changing the LED brightness if we've got little dial or knob, or we can simply move the pondweed further away or closer to the bulb. So the closer the bulb is, the higher the light intensity is. Another change you can make to the setup, which you could be familiar with, is to just leave the test tube open, so not sealed, and instead you just count the number of oxygen bubbles instead. Now, here's an example of the method. First, we measure a set volume of water into a boiling tube using a measuring cylinder. So that's like 40 cubic centimeters. So we control how much water is used. Then we place a 10 centimeter long strip of pondweeds, so we control the same plant species, same amount of chlorophyll as well, into the test tube or boiling tube, and then submerge it. Then we also add the sodium bicarbonate to control this carbon dioxide level. The next step is to switch on the LED when it's 10 centimeters away from the beaker containing the pondweed, and we want to wait 3 minutes. This is because we need to wait for the plant to slowly adjust itself, perhaps, to reach a stable rate of photosynthesis. And then, after those 3 minutes, we start a timer and start counting the number of oxygen bubbles produced. Or we can also collect the oxygen gas into a gas syringe and then just measure the volume after 2 minutes. And then the rate of photosynthesis is simply the volume of the gas produced or the number of bubbles produced per minute. And we record this. And then we repeat steps 2 to 3, but this time we move the LED bulb further away to reduce the light intensity, or just simply turn it down. So, for example, we could repeat this at distances further away, such as 20 centimeters, 30 centimeters, and 40 and 50. And you just need to list enough of them to show that you're repeating. And then you could even repeat the whole investigation a few times. And this is like any other practical in science. It's so we can calculate an average and ignore any anomalous results to make sure our results are valid and reproducible. And lastly, we create a graph. We will link the rate of photosynthesis to the distance from the light source. Now, after the practical, you'll plot a graph, as said, if you had light intensity on the x-axis and rate of photosynthesis on the y-axis, it would look like this. Now, you can see the line starts off straight, but then it starts to curve off and becomes flatter. So when the line is flat, the higher light intensity no longer increases the rate of photosynthesis because something else is perhaps limiting the rate of photosynthesis. Something is stopping it from achieving a higher rate of photosynthesis. For example, that could be another factor such as carbon dioxide. If we instead added more carbon dioxide, the rate of photosynthesis would rise again. The thing is, anyway, the other factors are controlled in this investigation. So you could also label the graph in a few ways. So whenever the line is not flat, light intensity is the limiting factor. Whereas when the line is flat, another factor limits the rate of photosynthesis. Now that's it for Biology Paper 1. I hope you found this walkthrough useful, where I sifted through all the advanced information for you to use. Good luck in your exams. Biology Paper 1 is going to be on the 17th of May 2022. Goodbye.